Good afternoon, students. Today, let us learn about certain experiments that are proving the absorption and conduction of water in plants. The experiment number 1 to show that root absorb water. Here, two test tubes are taken, test tube A and B. Here, A is the control experiment. In that test tube, it does not contain any plant, only water, and on its surface oil is present in test tube A. In test tube B, a small plant with a root is taken and it is introduced into the test tube B. After 2 or 3 days, we can see that in test tube A that does not have a plant inside, the water will remain in the same level after 2 or 3 days. But in test tube B, we can see the reduction in water level. After 1 or 2 days, the level of water is shown in the test tube B. What will be the condition of water is shown here. In test tube B, what will be the condition? Water level will come lower. So from that we can understand that water lost from test tube B because root absorbed water from B and that escaped water molecules by evaporation, it escaped through the leaves. That is why the reduction in water level. But in test tube A, water will remain the same. Again, you can see experiment 2. Here, it is to show that water is conducted upward through the xylem. The water will not be absorbed through everywhere in the plant, but only through the xylem tissues it is conducted upward. Here also in this, here in figure number 4.14, you can see that the water is coming. The red colored arrow, it shows that from the root hair the water is absorbed and cell to cell osmosis takes place and finally it reaches the xylem vessel, then only the upward conduction of water happening. You can prove the same experiment with a soft stemmed plant like balsam, or in our area we usually take this experiment for that മഷിത്തണ്ട് ചെടി അല്ലെങ്കിൽ കോലുമഷി എന്ന് പറയുന്ന ചെടിയാണ് യൂഷ്വലി വി ടേക്ക് ഫോർ ദിസ് എക്സ്പെരിമെൻറ്റ്. One English name is for that. I am not remembering it, so I am not telling it. Now you think it as a balsam plant. It is a soft stemmed plant. കോലുമഷി അതിനകത്താണ് we can see through the stem small lines we can see. The red lines are going. The xylem vessels we can see because its stem is somewhat it is translucent. So we can see the xylem vessels with the naked eye. If we take the cross section, we can clearly the xylem tissue, the stained pink color you can see. Here in the beaker, eosin dye is added to the water. Red colored eosin dye, that are the dye used in laboratories. In olden days, when we were at school time, we have done the experiment with red ink or blue ink we can use in the beaker instead of eosin dye. That also show the same effect. If we cut a transverse section, if we observe it through the microscope, we can more clearly we can see that the xylem vessel. So the experiment is proving that the water is conducted upward only through the xylem vessels. Then again the another experiment you see. Then one thing you see that blue box here, vascular bundle in the stem, root, leaves, stalk and leaf veins are all continuous and form an unbroken system of tubes. That vascular bundle meaning a sheath that covering the xylem and phloem vessels. Usually xylem run through the inner area and phloem run through the outer area of the stem. So that xylem and phloem together make the vascular bundle. Collectively they form the transport system throughout the entire plant. Water and salt travel upward in the xylem and food substances travel up and down in the phloem vessels. There are xylem and phloem. You learned about the conductive tissue. That xylem vessels usually run through the inner part of the stem and the phloem vessels usually run through the surface area. Here one experiment 3. Experiment 3, conduction of water through the xylem vessel. Here two in the experimental setup, two A and B, the two beakers are taken and the A twig with the phloem removed and xylem intact. In experimental setup A twig with the phloem is there. Phloem removed and xylem intact. I told you that xylem go through the inner part and the phloem is removed by the process known as ringing, experiment also called girdling. That process removing the outer surface of the stem, that is the girdling, that is known as the girdling. And so that the xylem will remain in the experimental setup and phloem is removed in A. In the twig A, you can see that. Then after 2 or 3 days, we can see that the leaf in that experimental setup A will remain healthy because the leaves get enough water through the xylem. The stem is absorbing. Through the stem also some water can be absorbed. You will learn which are the forces that are conducting water upward. So if we keep the stem, the tip of the stem submerged in water, it can absorb some water through it. So through the xylem vessel, water and minerals are reaching the leaves. So in the experimental setup A, the leaves remain healthy. But in B, you can see the xylem part is removed and phloem is present there. So the upward conduction of water and minerals is not taking place in B, so that the leaves is drooping and seem to be wilted. So the experiment proved that the conduction of water and minerals are taking through the xylem vessel. If in the plant setup B, xylem removed, so the leaf are becoming wilted. Here another experiment is here. Experiment number 4 to show that experiment number 4 to show that food from the leaves is conducted downward through the phloem in the stem. Here is also the same process, girdling is taken. Girdling, the same. The xylem vessel is there. So girdling process is done in experimental setup A. Then after 1 week or 4 or after 5 or 7 days, keeping the plant like that, we can see that the leaves remain healthy because the leaves in the stem are becoming, or at the end of the experiments, the leaves remain healthy because the leaves in the upper part of the stem getting enough water and minerals through the xylem vessels. Then again the part below the girdling, that part become shrunken, that shrink and begin to die, or that part shows unhealthy features. And you can see that the food that is made in the part of the leaves is flowing downward and it is showing some accumulation. The stem swollen with the accumulated food, you can see. when in the picture B it is shown clearly at the starting of the experiment the shape of the stem is shown. At the end of the experiment, that part above the girdling, that part becomes swollen because of the accumulation of food. Today I am finishing the lesson now, and I shall complete the progress check and other note for you, and I shall send it for you. Thank you for listening the class.